Hello, I'm Jeanette Hellgren Kotaleski, and I will introduce dBRAIN, which stands for Digitizing Brain Data for Health and Disease. And here at the bottom, you see all the senior researchers that are involved. So we come from different environments at KTH, but also from Karolinska Institute and Karolinska Hospital. So the reason why we started dBRAIN is, of course, that brain disorders are a big burden. They account for over a third of the healthcare cost in the Western world, and this is rising as we are becoming more aging population. But fortunately, we have developed a lot of new technologies, neuro tools, so we can also measure get a lot of data from both the healthy and diseased brain. And if we then use that data wisely, we can receive additional insights if we use models and simulations and AI, machine learning, and so on. So dBRAIN stands on three legs. It's data, models, and software. As said, it's a multidisciplinary collaborations between the three KTH schools and also Karolinska Institute and Karolinska Hospital. And we use AI-driven data analysis together with modeling and simulation to extract more sort of patterns and knowledge from existing data. And then we want to apply this result to improve diagnosis, prognosis, stratification, treatments, and so on the most common neurodegenerative disorders, Alzheimer's and Parkinson's, but also age-related tremor. So this is a typical situation that if many of us will be ill, get a brain disease, and then we go to the doctor that takes some tests and give us a diagnosis and treatment plan. What we want to do within dBRAIN is to add to this so that the individual patient data can be seen in a larger context together with additional data, and this so that we perhaps better can stratify patients in the future. So for that, we use AI-based data analysis, and when it comes to AI and similar things, we typically can discover new patterns in data. But these patterns might just be correlations to the disease. And if we want to understand better also the causal chain of events and the mechanism, then we also have to put our knowledge and construct models at different levels of biological scales, as we will illustrate. So key challenges in brain science in general, I would say, is to understand the relationship between structure and function, or structure and dynamics within local networks and between brain regions and so on. And another big challenge, if you want to understand the diseased brain or the aging brain, is to understand how slow changes can lead to changes in brain function and brain dynamics. And as I said, we focus on Alzheimer, Parkinson's, and tremor. And to address this issue, we use digital tools, obviously, modeling, simulations, and AI. And we do this at different biological scales, because we have data from all these scales, of course. So the first example relate to a quite detailed level of single neurons. So in Parkinson's disease, it's known that if a healthy neuron looks like this, then during the course of the disease, these neurons start to lose these distal dendrites. So it sort of shrinks, it loses some of the complexity. And the consequences, what is the consequences for the network then? This we can address using a pipeline we have developed, how we can build microcircuit level models by predicting where neuron comes close to each other, where they connect. And this we can do in the control network. This shows the connectivity probability between two neighboring neurons over increasing distance. And this is the same thing. And as you can see, the connectivity has decreased a lot for these more severe Parkinson's disease stages. So we can also quantify this in a more sort of rigorous manner and look at something called clicks. There we use topological data analysis. So this is a sort of different motives, how cells can be coupled. And if we explore and search for these clicks in these networks that we build, then we can see that for the control stage we have many more different types of click represented. But then when we progress in these disease stages, then this variability decreases. And what is really interesting is that if we move interneurons, it really shrinks. We can also look at these different motives and ask what kind of neuron types build up these motives. And then we can see that this changes quite dramatic over different disease stages, and especially for these more complex clicks, we lose the variability of neurons a lot. So now we will progress towards the next scale, sort of towards the mesoscale.
My name is Wojciech Chachulski, and I'm a professor at the math department at KTH. I am specializing in geometry and geometrical methods in data analysis, and my role in dBRAIN is to provide a technical support regarding the use of these methods. You might ask, what connects geometry and data analysis? It turns out that many analytic methods, for example, PCA, clustering, SVM, and other kernel methods, they all use geometry to extract information from data. One common step in any geometrical methods is to represent data in form of a space. The reason we need data analysis is to make sense of data whose complexity prevents us to make conclusions about that data and its formation. Data analysis is about simplifying. The role of a spatial representation of data is, however, not to simplify. These spaces are often as complex as the data itself. The reason we like to have a spatial representation of data is because it allows us to extract various invariants, such as simple counting statistics. Jeanette already mentioned a use of such counting methods where we count various clicks to understand networks related to stages of Parkinson's disease. Another important invariant that spatial representation allows us to extract is homology. We can use it, for example, to subgroup Parkinson's patients based on a correlation information extracted from fMRI images, which is a joint project with Joanna Pereira's group at Karolinska.
Hi, my name is Arvind Kumar, and I'm one of the PIs in the dBRAIN project. To better understand how neural activity and circuits are changed across the brain in PD, we analyze the MEG signal. Instead of analyzing the oscillations, we focused on how the power of the signal changes as a function of its frequency. This is something we can quantify as this parameter lambda. And we are interested in lambda because it is related to the excitation and inhibition balance in the underlying neural circuits. We found that on average, PD patients, they have higher lambda than healthy controls, and higher lambda corresponds to higher degree of inhibition. So in this case, PD patients have higher inhibition in sensory and motor regions compared to the healthy controls. Surprisingly, we also found that medications such as levodopa do not influence this particular parameter. We are very much optimistic that we can further develop this measurement and develop lambda as a new biomarker that can be used in clinical practice. Besides analyzing the data, we are also modeling brain circuits to understand the origin of pathological activity. One such pathological activity is this change in the shape of the transient response of the basal ganglia. Normal response looks like what is shown in the blue, and the pathological response is shown in the red. We use the network model to understand which connections in the network are responsible for this change. To represent the transient response, we define 18 parameters, and now we can draw the healthy state of the transient response as these green circles. The pathological state is shown in the red, and the response of the network, once we have changed certain connection, is shown in blue. We can see, comparing these 4 panels, that when we change this particular connection from D2 to TI, we can actually recover pretty much the healthy state of the transient response as shown in the overlap between blue and the green curve. This suggests that this particular connection from D2 to TI neurons, this is a very crucial connection in shaping up the pathological dynamics in Parkinson's disease.
My name is Rodrigo Moreno. So we are tackling the problem of brain connectivity at the macro scale, at the brain level. So we have been working with the structural connectivity problem. So we have developed a new method that takes as an input the tractogram of the image and then filter it. So then you are sure that you are getting the most reliable tracts that are representing the neurons connecting the different regions of the brain. So we're currently applying this method to classify patients and controls, patients of Alzheimer's. And then our idea is to connect this to explainable AI methods to unravel the causes of the disease that can help doctors to come up with better treatments. We have been also working on synthetic MRI aging. So the idea is that we have one image of one subject, in this case 51 years old, and we generate synthetic images of the same subject at different ages. So the idea of this method is to use these new images to create templates of brains at different ages, and then to compare them with patients of Alzheimer's. So that way we can see the status of this patient with comparing it with healthy population.
Moving over to our work relating to behavior, my name is Erik Franzén, and I'm a professor of computer science at KTH. In one of the projects, we're working with medical doctors at the Danderyd Hospital in the clinic where they have tremor patients. We have developed a mobile phone app to measure the muscle shaking of these patients. The goal is to be able to easily monitor the patient, assist in diagnosis, and possibly evaluate medication. What we have done is to produce this mobile phone app that can be strapped to the patient, and also we're developing the different computational analysis of the signals from the muscle shaking. In another project, we're working with Per Svenningsson at the Karolinska sjukhuset department where they have Parkinson patients. In this case, we're using machine learning to analyze data from eye tracking experiments. The goal here is to find biomarkers, patterns of eye movements that differ in the patients from the healthy individual, to be able to assist in a diagnosis and possibly in prognosis.